We made it to the last talk of the day, and I have good news for you. It's the last talk, and I talk really fast, and it's still sunny outside, I think. So let's go. So I'm Sarah Munro, and my co-author for this presentation is Jeremy Chacon. He's here in the room. We are bioinformatics analysts at MSI, the Minnesota Supercomputing Institute. My goal here today is to make the case for why defining distinct cell groups in single RNA sequencing analysis is so challenging, and to present some of the recommendations that we have developed for mitigating bias in our work. And I'll preface this by giving you a disclaimer that we don't have all the answers. There is not a one-size-fits-all solution for cell type annotation. We've seen lots of different data sets, and it can be quite challenging. But we do have a lot of practical experience, and so we're trying to work toward best practices. So what is our experience? Collectively, the analysts within MSI Bioinformatics group have analyzed over 100 single cell projects. And we also do a lot of other work too, not just single cell. The majority of these projects have been with mouse and human cells, but we've also had other species such as turkey, rat, pig, dog, and rhesus macaque. And today I'm going to share you 3 example projects where we employed strategies to limit bias in cell type annotation. But first, let's talk about why cell type annotation is challenging. So this is a very simplified single-cell RNA-seq workflow. We start with FASTQs and want to generate a cell-by-gene count matrix. And then this cell-by-gene count matrix needs to be processed and annotated with cell groups related to the experimental design, and then we can proceed to downstream analysis and learn some cool new biology. This part of the process where we create the annotated cell-by-gene matrix is actually quite complex, as you've heard in a lot of the other talks, and requires multiple steps. Quality control, normalization, dimensionality reduction. The piece I'm going to focus on today is cell type annotation. So I've divided it into 2 general areas, right? So there is a reference-free method for cell type annotation, and then a reference-based method. And the reference-free approach is something that we've been doing for quite some time. It can be applied to any organism or cell population. It's really useful if you want to identify novel cell subtypes, and it's what we used before we had a lot of reference data available to us. The reference-based approach generally works best for model organisms where you have known cell types that you want to look at. Clustering is required for reference-free approaches, but you don't have to do clustering if you're doing a reference-based annotation method. And with reference-free approaches, you also have to have appropriate marker genes that you can use for your annotation, whereas with a reference-based approach, you don't need to have any knowledge of specific cell type marker genes, but you do need to carefully choose what your references are. And I'll talk to you about that. So, the challenge that I presented is that we want to maintain objectivity and avoid bias when we're doing this cell annotation process. And there are several sources of bias that can come into play here, and I'll go through each of them in the context of the the reference-free or the reference-based approach. So with reference-free annotation, the one source of bias that we don't have to consider is reference data. We aren't choosing what that would be. But we do have to be concerned with things like cluster resolution. This can be quite challenging. You want to strike a balance between overclustering, where the cell type differences are simply due to noise. Remember, this is all just based on gene expression. And underclustering, where we might be missing new cell types that we want to be able to understand with our downstream analysis. The next thing we have to consider is avoiding misleading visualizations, such as UMAPs, which we are all familiar with. The use of UMAPs is still somewhat ingrained in the field. We still use them, but we try to avoid them in our, de-emphasize their use in our data interpretation so that we don't misinterpret the distances on UMAP plots. Then we also want to be careful when we're doing one versus all DE tests. And what I mean by this is we'll typically take our different clusters and we'll take one cluster and do differential expression comparison of that cluster against all the other clusters in our data set and try to look for the genes that are different, right? And we call these our top gene markers. And so one problem with doing this is that this can lead us to a form of what we call double dipping, where we essentially use statistical testing to form our groups in our data set, and then we do a second set of statistical testing on those same groups to answer our biological hypotheses. And this can be problematic because it can lead to inflated type 1 error. Now, in reference-based annotation, you don't have to be concerned with your choice of cluster resolution or using DE testing to find top gene markers, but you do need to still be concerned with overinterpretation of UMAP visualizations, and you have to be careful about your selection of reference data. So if you are too specific in terms of the reference data that you choose, you may misannotate contaminating cells as the cells that you're actually interested in. If you're too broad, you may miss finding novel cell types. So let's get into some examples. So this first project that I'll show to you was a project where we used reference-free annotation of mouse cell mixtures of known cell types. So in this project, the PI created defined cell mixtures with 3 sorted cell types, and therefore he could choose a reference-free approach and wasn't worried about doing any sort of clustering or doing any sort of reference-based approach because he knew which markers he was going to use for annotation. To avoid UMAP visualizations, we started with a cluster tree diagram, which is a visualization tool and R package that's been available for many years. It was published originally by Zappia and Ashlock. So in this cluster tree, you can see that each cluster resolution is a different color. For this project, I swept through Louvain cluster resolutions from 0.1 to 1.0 in increments of 0.1. The size of the points indicates the number of cells in each cluster resolution. And then the arrows indicate the number of cells moving to a cluster at a higher resolution. You can see some clusters split as the cluster resolution increases. And as you get to very high resolution, you may see a lot of cells switching back and forth between cluster lineages. Other clusters are highly stable across resolutions. What's great about this figure is that you can see all cluster resolutions in one plot, and you don't have to look at a UMAP and potentially misinterpret cluster similarity or relatedness based on where the clusters are on your UMAP plot. So for this project, I was interested in figuring out a good metric for choosing cluster resolutions for the PI to use for his expert annotation. And so this led me to the SC3 stability metric. So this is the same cluster figure, now colored based on the SC3 stability metric value. This metric for evaluating cluster resolutions across a range of resolutions was originally developed by Kiselev and colleagues. And each cluster is scored based on how many cells they share with other clusters at different resolutions. Values close to 1 indicate the greatest stability. And you can see this illustrated in the darker red clusters that are highly stable across multiple resolutions. However, I found it to be challenging to interpret color scale across this very large complex tree diagram. So I decided to try a different approach to visualize the SC3 stability metric across different cluster resolutions. And so I used simple box plots. Sometimes simple works. So I did some box plots of the SC3 stability values by cluster resolution. The size of each of these open circle points represents the number of cells in a given cluster, because I thought it would be interesting to see those. And then using this visualization, I was able to nominate resolution 0.3 here and 0.4 as the 2 most stable cluster resolutions based on the median SC3 stability value for those cluster resolutions. Great. So then I passed this on to the PI, and then he went onward to do some expert annotation via marker gene expression for resolution 0.4, which is the bottom green set of clusters. And then he ultimately defined 3 cell types, the main cell types that he was interested in, and then other immune cell types and contaminating cells were in the data set. And one interesting part of this is even though he was looking at resolution 0.4, he ultimately decided to merge these 2 clusters into this cluster and call them endothelial cells. And so the final annotation was equivalent to resolution 0.3, which was the most stable resolution. So in that project, we tried to avoid UMAPs as much as possible and use something more concrete for doing cluster resolution comparisons. Moving on to example 2, this project was a reference-based annotation of mouse hippocampus. So this was a pilot project with one male and one female mouse brain sample. And initially I did reference-free based approach like I did for the other project, but the PI decided to pivot to using brain references because it would be too complicated to do expert annotation of all of the cells in his data set. And so I started pulling references. The initial references that I tried using are shown here. I used single R as the tool for doing the annotation. And I pulled a mouse bulk RNA-seq data set that's available. And then I pulled the CZI cell by gene whole brain data set. And because it was brain tissue, we found a lot of neurons, great, but we actually wanted very specific neuron types because these were actually hippocampus samples. They weren't just brain. So we moved on and found a different reference from CZI cell by gene that was hippocampus specific single cell data. And what was really great about this data set was that they provided cell ontology terms. And I would encourage people, if you are doing single cell RNA-seq analysis, to try to use this controlled ontology, because then we can all use the same labels for our cells and not be inventing different cell type labels. And what's really cool about this too is that you can use R packages such as ontoproc and ontology plot to create hierarchical trees to show the relationship between different cell types. And so with this data set, we were able to identify 8 different neuron cell types. I apologize, you can't see everything, but there are different neuron cell types, but these weren't actually the labels that the PI was interested in. So we moved on to one final annotation, which the PI found. It was from Genelia Farms' HippoSeq Atlas. So it's actually an atlas of bulk RNA-seq data from laser capture microdissection from hippocampus. And so, you know, even if the technology is old and it's been around for a while, it can still be really, really useful. And so we looked at the 2 different data sets that were available. The researcher said, hey, I think that this is really useful because it's getting the specific regions of the hippocampus that I'm interested in. And so we did both of them, but this was the one that he was interested in pursuing. We looked at some marker gene expression, CAMK2 alpha, and also compared the final cell counts between the samples to make sure that we weren't missing anything. So we ended up with an annotation for that project. Now, in example 3, our final example, we were doing, this is actually Jeremy's project. Jeremy did reference-based annotation of human immune cells from multiple tissues. So the PI for this project extracted immune cells from spleen, colon, liver, and small intestine. And right from the outset, he said, I do not want to do any expert annotation. I want to use reference-based approaches. Let's make it easy on ourselves. Great, okay. So Jeremy ultimately ended up using 4 different reference annotations. They're listed out here, and I'll go through the details of them in a moment. The tools that he used were SC-type and single R. And he started with the SC-type immune-only reference, thinking that that would be the way to do this project. But found that there were some questionable annotations in that data set, and so he decided to try using the SC-type immune plus the tissue type combined reference. And so for the liver tissue, Jeremy constructed this alluvial plot to show how much agreement there was between the 2 cell type references. And so he's highlighted these traces in green, these paths between the data sets. So these are the memory CD8 positive T cells, which are getting annotated as CD8 positive NKT-like cells, and also immature B cells here in the SC-type immune-only reference. Whereas with the additional combined data from liver tissue, they're getting some slightly different results. But generally, everything is an immune cell, and there is a lot of overlap. Neutrophils are consistently called neutrophils between the 2 references. Great, this worked well for liver tissue, but there's more. Jeremy moved on to look at the colon tissue using the same 2 references and found that a lot of the cells were annotated as cancer cells, and those are highlighted here in red. And what Jeremy was showing with these paths here is the cancer cells that are then annotated as immune cells or other cell types in the SC type immune only reference, which is problematic. But there were some cells, the paths that are shown here in green, that were in agreement between the two data sets. So there was this similar issue with the small intestine data set as well. And the spleen data was actually quite clean, just like the liver data set. So after using these two references, Jeremy pulled in two more references, which I mentioned initially, and tried to compare them using this multi-panel alluvial plot. We're actually not including the immune-only reference. We're including ImGen here on the left, SC type combined here, the single-R mouse bulk RNA-seq data set that I used in my previous project, and then we're also including single-R ImGen data again here, so that you can see the comparison between this and the bulk RNA-seq mouse tissue. And you can see again that the cell types are getting misannotated from multiple references. And so what they ultimately ended up doing was using the SC type combined reference and excluding cancer cells that were labeled as cancer cells for their downstream analysis. All right, so to summarize, I've tried to show you today some of the sources of bias that come into play when we're doing cell type annotation, things that we're concerned about. Cluster resolution, avoiding UMAP visualizations, trying to not do a ton of DE testing before we want to do our final DE testing, carefully choosing reference data. And then we have some recommendations here. So for reference-free methods, the two main recommendations are to try to visualize cell clusters with cluster diagrams and choosing the top cluster resolution with SC3 stability metric plots. For reference-based annotations, you want to carefully choose multiple annotation references. Don't just choose one because it might not be the one that best matches your data. And that using alluvial diagrams to compare annotations is really useful. So with that, I will acknowledge the PIs whose data we are showing in this presentation that we worked on, and then many of my colleagues at MSI who have had a lot of single cell analysis conversations with Jeremy and I. I want to call out that Marissa has a really awesome poster here at GLBIO. She was presenting it today. It's still going to be up tomorrow. She's got a lot of really interesting vignettes about single cell analysis challenges. And then Natalia is presenting next week at the StatGen conference. And we're done.
Thank you so much, Sarah. Any questions?
Online.
Yeah, this is an online question. So for your example one, have you compared results with UMAP, at least to assess how insightful they can be in such a case when compared to the SC3 stability metric? Can the UMAP lead to the 0.3 resolution?
I am not following that question. Maybe.
Okay.
Can I read it?
For example one, have you compared results with UMAP, at least to assess how insightful they can be in such a case when compared to the SC3 stability metric? Can the UMAP lead to the 0.3 resolution?
I mean, yes, you could. I suppose you could use the UMAP to lead yourself to a 0.3 resolution. I think that you would have to do the one versus all marker gene analysis or spend a lot of time looking at marker genes first. I mean, you could just start with that. I think the challenge there is deciding how far up the tree to go and where to start on the tree. So yeah, I guess you could, but we did not compare that.
Great talk. So right now you're talking about mapping single cell onto single cell. Have you tried mapping single cell onto image-based spatial transcriptomics yet?
So we do a lot of spatial mix projects in our group. I have not personally done that. Has anyone else done that yet? No, yeah. So there's 4 of us here, but none of us have actually done that in our group. But yes, it is possible to do that as well.
Okay,
Yep.
cool. Thank you.